Hello everyone. We will wait for a few more minutes so that people can join. I guess I can start now. Hello everyone. My name is Himanshi Singh. I'm a PhD candidate at BSB department in IIT Kanpur. So we will be discussing some of the material covered in week 6, and then we'll go back to some of the previous year assignment questions, which will help you in solving your current assignments, and it will help you also in getting some of the insights of the concepts which were taught in the class. So let's go back to one of the lectures which Professor Mandal has discussed. I hope you guys are able to see my screen. So this was the first lecture which was covered by Professor Mandal in week 6, and the title of the talk was Energy Production from Organic Waste through Anaerobic Digestion. So till now, we have covered thermochemical processes such as pyrolysis, combustion, and all those things. Those were thermochemical processes. From here, we are discussing about the biological routes for converting waste into energy or getting some of the commercially important products from the waste material. So in this current slide or in this current lecture, we will discuss these points which are marked in red. First, we will discuss about what exactly are anaerobic digestion, specifically for biogas production, mechanism of anaerobic digestion, microorganisms for anaerobic digestion, because as I mentioned that this is a biological route for converting waste into useful energy or products. Hence, some of the microorganisms which are associated with anaerobic digestion. Next, pathways for anaerobic digestion. We will see what are some of the pathways through which we are getting energy from the waste material. Then next, the pre-treatment of lignocellulosic biomass and waste. It is a special class of waste and it requires some of the pre-treatments, the materials which are high in lignocellulosic material, they require pre-treatment, and we will discuss some of the pre-treatment methods here. If we have time, before discussing assignment questions, we will also look the flow sheet for anaerobic digestion of waste, types of anaerobic digestion processes, kinetics of methane formation, anaerobic digester and their types, operation of anaerobic digester, two-stage digester and process, impurities in biogas and their impacts, sludge volume in a digester, sorry. So first we'll look at these red points. So basically, what is anaerobic digestion or feedstock and the primary products which are involved in anaerobic digestion? Basically, anaerobic digestion is a series of biological processes in which microorganisms break down biodegradable material in the absence of oxygen, right? So here we are using the natural occurring microbes which have the capacity to degrade some of the material to produce energy, right? So we do not need to input any external heat source, or these operations are not performed at very high temperature. So we do not need to worry about external energy supply in such kind of operations. So basically, what is the input for anaerobic digestion? So we can either have energy crops, animal waste, industrial waste, municipal solid waste, restaurant food or other types of food waste, sludge from waste water treatment plants, right? So these all are the inputs for the anaerobic digestion. And from the anaerobic digestion, what we will get some of the products such as biogas, which is the most important product. It is basically a methane containing gas, also called gobar gas. Then we will get hydrogen, we will get biofertilizer, we get compost, and we also get some of the reusable water. Hence, this is comparatively, it's an less energy intensive process, but still it requires some energy for its operation purposes. So as I mentioned, that the basic or the primary product which we go for anaerobic digestion is the biogas. So what is a biogas? A biogas is a combustible mixture of gases. It consists mainly of methane, as I mentioned earlier, and carbon dioxide, and is formed from the anaerobic bacterial decomposition of organic compounds, especially in the absence of oxygen, right? So the major constituents of biogas, though the major component is methane and carbon dioxide, there are other components present in the gas, because as we know that these are the input materials, right? This is the input feedstock. So all of it won't be converted completely into methane or carbon dioxide. So there will be some intermediate products as well as some of the things which will be present in the end product. So what are those things? So methane, obviously, it is more than 50% of the total biogas. Carbon dioxide, again, ranging around 25 to 50%. Other constituents, apart from the major component that is methane and carbon dioxide, is nitrogen, hydrogen, hydrogen sulfide, and oxygen, though in very less percentage, but still they are present in the gas. And we should know this because by knowing the components present in biogas, we will be able to generate or make some operations or some processes on the biogas produced for its purification purposes, right? So some specific biogas production potential and methane content for several waste. This is some experimental based data. It will vary and it will completely depend upon the conditions in which you are calculating these units. Also, the type of raw material you are inputting for the anaerobic digestion process. For example, if I'm saying wheat straws, even in the wheat straws, there are different varieties of it, difference in the constituent of wheat within the wheat straw. Hence, these percentage are just for you to understand and get some idea to what to expect from such waste products, right? So if we are having a wheat straw, the dry matter content, that is the percentage of dry matter in wheat straw, is approximately 86%. The biogas which is produced from this wheat straw, which is 86.5 percentage by weight, the biogas produced, that is in liter per kg from this substrate, is around 367, that is 367 liter per kg of this substrate. And the methane content in this biogas produced from this raw material is 78.5%, right? Similarly, you can interpret this for every single waste material. You have to see that depending upon the waste material, the biogas potential as well as the methane present in the biogas varies. Why this varies? Because as I mentioned in the introductory slides, that there is a separate discussion for lignocellulosic material, right? So the material which are high in lignocellulosic content, it won't be so suitable for biogas production directly. Hence, if we are producing a biogas from a raw material which is high in lignocellulosic content without pre-treatment, first of all, it will yield very less biogas. And even in that biogas, it will have very less percentage of methane. So here, for example, let's take this example, which is less. For example, grass. Grass is there. Grass is basically 16% of weight, right? Out of the 16 percentage of solid weight, the potential for the biogas is 557 liter per kg of the substrate, and the methane content is 84%, which is comparatively very high in comparison to wheat straw. So you can decide which operation you want to perform based upon the raw material or the waste material you have with you. If you are dealing with certain agencies or institutes which have a continuous supply of dried leaves, right? For example, if they are very big campuses, they have trees, so they will shed their leaves. They don't know what to do with it. For them, it's a waste. But if you are there at the intermediate between the waste management and their waste disposal committee, what you can do is you can basically take the dried leaves and you can subject it to anaerobic digestion. At the end, you will get methane or biogas as a end product, which you can further use for other purposes, right? So in continuing our discussion, these are again some of the other type of waste or raw material which are subjected for anaerobic digestion, and their methane content and biogas potential liter per kg is given here. Here you can see that the horse dung, the cattle manure, these two have very high biogas potential, right? Because innately cattle manure is very rich in methane content. And if we are subjecting that to the anaerobic digestion, we are enhancing its further potential, right? So, and also you can make certain comparisons looking at these tables. For example, though the potential of biogas production is very high in cattle manure, but the content of methane is comparatively lesser than for say like even in this table for say with beet leaves. Though in beet leaves the biogas potential is very high, but the methane content is also high, right? If we look for other examples, for example, it has 480 liter per kg of substrate potential for making biogas, but it only has 60% of the methane content. This 480 is greater than cattle manure, right? But their methane content is approximately same, right? The highest value for cattle manure is 60, while in the pig manure it is 60. Hence, you cannot estimate based upon only the waste material which you have. If your end goal or the end aim is to produce methane, and biogas is one of the product which has methane, right? Then you need to go for substrates or waste material which have high methane content, irrespective of their biogas potential production, right? So we have seen some of the like basic examples of what to expect when we are subjecting these type of raw materials to anaerobic digestion, what amount of biogas we can expect or what amount of methane we can expect, right? Now we will look at the steps which are involved in the anaerobic digestion. So specifically, as I mentioned, that for lignocellulosic material, you need pre-treatment, right? So we will discuss further in the pre-treatment unit that what are the pre-treatment options available to us for treating lignocellulosic material. So general steps of anaerobic digestion includes that we have complex organic compounds, right? We have the waste which we have like cattle manure or leaves or wheat straw. They are very high in carbohydrates, fats, and proteins. So the first step is with the help of hydrolytic bacteria or enzymes. It depends upon whether you are using microbes directly or you are using the enzymes extracted from these microbes. Hence, both can work. So hydrolytic bacteria or enzymes, the first step is hydrolysis, which will form simple organic compounds from these carbohydrates. As you must be knowing that carbohydrates and fats and proteins, they are polymers, right? Carbohydrate is a polymer of sugar or a monosaccharide. Fats are polymers of fatty acids. Proteins are polymers of amino acids, right? So if you're hydrolyzing these complex organic compounds with the help of enzymes, it will break these complex and larger compounds into simple, smaller subunits of these compounds. So for carbohydrates, it is sugar. For proteins, it is amino acid. And if you are breaking larger proteins, but not into their constituent amino acid, you get peptides. And for fats, you will get fatty acid, right? So after getting these simple organic compounds, then you subject these compounds to acidogenesis. Again, here we are using some of the microbes which will be able to decompose or metabolize these small organic compounds into short chain fatty acids, right? These are basically fermentative bacteria. Here is the role of anaerobic digestion, right? We are not subjecting it to aerobic digestion, we are subjecting it to anaerobic digestion, and it is called fermentation. So when we subject small organic compounds to fermentation or the microbes which depends upon fermentation for their energy usage, they are called fermentative bacteria. They convert these small compounds into short chain fatty acids. Now, these short chain fatty acids further can be metabolized or converted into acetate or carbon dioxide or hydrogen. It depends upon the type of bacteria you're using. If you're using acetogenic bacteria, it will produce CO2 and H2. If you're using acetogenesis or bacteria which produce acetate, they will produce acetic acid or acetate in short. Further, both H2CO2 and acetate can be converted into methane and CO2, which is our biogas, right? So if we are converting CO2 and H2 into methane and CO2, it is the role of methane producing bacteria. We will discuss in the coming slides who are those and what are they. Then if we are converting acetic acid or acetate into biogas, this process is called methanogenesis. From the name, we can guess what the process might be. For example, methanogenesis means methane producing bacteria, right? They are producing, genesis means making. So they are producing methane by using substrates such as acetate, right? So these were some of the basic steps which are involved in anaerobic digestion. Here we will see, despite having multiple approaches to get methane out of the complex organic solid waste, we have some designated percentages or the amount for which we can decide whether our process is effective or not, or at least to get an idea that what at the end of the day our product or the waste material is getting converted into. So for example, if you have 100% of organic compounds, right, and you are subjecting it to hydrolysis and acidogenesis, as you have seen here, that this is the hydrolysis step and this is the acidogenesis step. After this step, almost 76% of the organic compound or the waste material which you have taken is converted into short chain fatty acids, right, with the help of microbes. Out of that 100%, second largest constituent goes to the formation of acetic acid. And last but not the least, some part of organic compounds is converted into hydrogen also, right? Now, these short chain fatty acids, as I mentioned, they are subjected to fermentation. These, under the fermentative condition, almost more than 50% of the simple constituents or simple fatty acids or short chain fatty acids are converted into acetic acid, while around 24% or less than 50% is converted directly into hydrogen. Now we subject the hydrogen CO2 or the acetic acid to different types of microbes, either the methanogenic type of microbes or other type of microbes, to make methane, and the process is called methanogenesis. So if we have acetic acid and we are subjecting it to microbes which feed upon acetic acid to make methane, they are very effective and they convert almost 72% of the acetic acid into methane. While if you are subjecting microbes to combine hydrogen and CO2 for making methane, they are less efficient but still bit comparable to the second type of micro, to the first type of microbes, and they almost convert 28% of the available hydrogen into methane, right? So these are the three basic stages of anaerobic digestion: the hydrolysis and acidogenesis, which makes short chain fatty acids; acetogenesis and dehydrogenation, which helps in either metabolizing the acetic acid or combining the hydrogen with carbon dioxide to make methane. And the process of making methane is called methanogenesis, right? Then after this, we have elementary description of AD. AD here is anaerobic digestion. I would like to stop. I see Arun. Hi, Arun. Are you able to hear me? You can text me in the chat box. And just give a brief intro about you. Where are you from? What are you doing? And if you have any questions, do let me know. So here we will talk about the basic elementary description of aerobic digestion. As we have seen earlier also, that if we have a stoichiometric equation of a reaction, it helps us in understanding of how much of a raw material is getting converted into a desired product, or what type of uh, amount of energy or substituent catalysis or catalyst we need to provide to get the desired amount of product, right? So it helps us in giving us some of the overview of the whole process, which we are not able to see, right? Because everything is getting done inside the microbe. We don't know how much of carbon is getting inside the cell and it is getting converted into biogas, right? So we have some of the methods, and one such method is to get the stoichiometric reaction of the whole process, right? And one such reaction or the constituents of a reaction is given here. So in the case of chemical composition of the organic matter, the stoichiometric equation is given here. It is mentioned in this paper in 1991. It is widely accepted equation, right? As you can see, the CnHaOBnC is basically our raw material or the organic compounds, in the form of waste which we have, right? This is the amount of water which is required for the reaction to take place. This is the produced methane, carbon dioxide. As I mentioned, this is the biogas constituent. This is for the biomass. And why we are talking about biomass here? Because the microbes are already present in the reaction, right? So they will also have some of the mass, and they will be on the product side. This is the ammonium and this is the bicarbonate, right? So as you can see, basically these N, A, B, C, it depends upon the type of raw material you are having. What are the constituents of that raw material? This is the bacterial dry mass. At the end of the day, bacteria is also organic creature, hence its signature will be organic and it is represented like this. Some of the fractions or the amount of cells which convert waste are given here. So S is basically the fraction of waste converted into cells. E is the fraction of waste converted into methane for energy specifically. This is for the waste that is being digested, hence our raw material. And this is the bacterial dry mass, right, which is at the product end. Now, we have seen the basic steps for anaerobic digestion. We have seen one of the stoichiometric equation which guides the anaerobic digestion process. So at the end of the day, if we have a product, we need to know the yield. So yield is basically what amount of the substrate or here the waste is getting converted into yield, is getting converted into biogas or the product in general, right? So the factors which affect the yield of a biogas from a waste material are first, solid content in the feed waste, as I mentioned. The constituents which are present in the waste material at the end will determine the amount of biogas you are going to get from that product, right? We have seen some of the examples in the table here. So these are the waste and this is the methane content. So depending upon the type of waste which we will have different constituents of the methane or different content of the methane. Second is C to N ratios, that is carbon to nitrogen ratio. Third is temperature, because we are getting the energy or the biogas from the microbes, right? So the growth of microbes highly depend upon the temperature and pH in which they are subjected to, right? So if the temperature is not suitable for the acidifying bacteria or the methane producing bacteria, the bacteria are not going to work. Hence, our yield at the end of the day is going to be decreased. So it depends upon the temperature. pH of the solution. Again, as I mentioned, bacteria or the microbes, their growth, they are highly dependent upon the temperature and pH. So if you're altering these conditions, specifically pH and temperature, the microbes are not going to survive if it is not optimal for them. And hence, they are not going to do their job, which is basically converting waste into a useful product for us. That is biogas here. Next is the retention time. So retention time, obviously, retention time is a factor which highly affects the yield, not only in the anaerobic digestion, but all other processes, right? Then the type of digester, we will discuss further that there are different types of digester and each digester has a different preference for the material which they are able to convert into biogas. And last one is the microbes types, right? So if our raw material is very rich in fatty acids or small chain fatty acids, and we are subjecting it to bacteria which do not know how to utilize these acids, hence we are not going to get any biogas or any product out of it. So the selection and the type of microbes at each step is one of the crucial steps for getting a better yield at the end of the whole anaerobic digestion process. Next is the, as I said, that we're going to discuss some of the microbes and the groups because earlier we have discussed that we have fermentation, we have acid producing bacteria, we have methane producing bacteria, right? So to what groups they belong to and what are the different groups and the microbes which are present in those groups, right? So the first group is non-methane forming bacteria or microbes. The second group is methane forming bacteria or methane forming microbes. In non-methane forming microbes, you have two functions, which is first hydrolyzing and fermentative bacteria. Next is the acetogenic bacteria, right? In hydrolyzing and fermentative bacteria, there are two, they basically perform hydrolytic, that is hydrolysis, and acidogenic, that is they are acid producing bacteria. In acetogenic bacteria, they produce hydrogen from acetic acids or compounds which generate acetic acid. Then and next is the homoacetogen. We will discuss what are homoacetogens in the coming slides. Next group is the methane forming bacteria. These are the one which are directly producing biogas, right? From the material or the raw products or the end products of non-methane forming are the products for the methane forming bacteria. So methane forming bacteria, there are further two classes of methane forming bacteria or microbes. First is the acetoclastic bacteria, which are acetate degrading bacteria. Next is the hydrogenotropic microbe, which hydrogen utilizing bacteria. So if you remember in this, we have hydrogen as a product as well as acetic acid as a product. So the microbes which produce methane from acetic acid are called acetoclastic. The microbes which produce methane from hydrogen are called hydrogen-tropic, okay, or hydrogenotrophic microbes. I hope this is clear. So here we have two stages, right? For this stage will be non-methane producing bacteria. This stage is methane producing bacteria will be used. So in non-methane producing stage, we will use these type of bacteria which are hydrolyzing and acetogenic. While in the methane forming stage, we will use these two type of microbes which belongs to acetoclastic or hydrogenotrophic, right? So now we will see some of the roles involved in the main three stages of biogas production. And what are the three stages? The hydrolysis, fermentation, the decomposition into short fatty acids, and at the end, getting the products into biogas form or the methane form. So in the first stage that we use fermentative bacteria and also the ones which do not produce methane, right? So they are non-methane producing microbes. So in hydrolysis and fermentation of organic substances, what kind of substances we have? Or the microbes which feed on these components. First is cellulose decomposing bacteria, as feedstock and agricultural waste are very high in cellulose. Hence, we use these type of microbes which decompose cellulose. And to what they decompose, as I mentioned, these are the complex compounds, right? So cellulose is a carbohydrate, protein decomposing bacteria, and fat decomposing bacteria. So if you decompose cellulose or carbohydrate, you're gonna get monomers, oligomers, or saccharides. Saccharides are sugars. Monomers and oligomers are the smaller units of carbohydrate, right? So cellulose is a complex molecule. If you break it down, either you will get a monomer and this monomer will not be further be able to metabolize or digested. Oligomers, they are not complex, but they are made up of 3 to 10 monomers, right? And saccharides are basically sugars. Protein decomposing bacteria or microbes, if you use microbes which degrade or decompose protein, the monomer or the smallest unit of a protein is an amino acid, right? Hence, these microbes will convert protein into amino acids or peptides. As I mentioned earlier, that again, like oligomers, they are a chain of 2 to 10 monomers, right? Similarly, even peptides, these are 2 to 10 amino acids in a chain, and those are called peptides, small proteins. For fat, as I mentioned earlier also, that if you decompose fat, you will get fatty acids and alcohols. So these are the smallest amount or the monomers through which these complex compounds are made of. So hydrolysis and fermentation basically does what? It basically breaks down the complex molecules into smaller compounds or the smaller constituents of these complex compounds. Now, all these are what? These are short chain organic acids. right? Or aldehydes or H2 and CO2. Again, this depends completely upon the type of microbe. For example, there will be a microbe which is very efficient in decomposing a protein into its constituent, that is amino acid. But there might be some other enzymes or microbes which will be able to just shorten the protein into peptide. So these end products, it depends upon the type of these microbes you are using. Because this is a very generic class, cellulose decomposing bacteria, right? So it is a broader class. There are multiple microbes which will be under cellulose decomposing bacteria. So it depends upon what organism you are using, and based on this, you will get the end product. So that end product of this first stage is basically short chain organic acid. To summarize it, hydrolysis and fermentation, the complex molecules are getting converted into shorter chain compounds or shorter chain acids, right? Then this will yield to uh, this will act as a raw material for a second stage. So our second stage is basically hydrogen producing acetogenic bacteria, right? So what was hydrogen producing acetogenic bacteria? It basically decomposes the substance. As I mentioned, the end product of first stage is the initial product or the raw product for the second stage. So this will convert these microbes, these acetogenic bacteria will convert small chain fatty acids or organic acids or alcohols into acetic acid, H2 and CO2, right? Now, what we have here, we have acetic acid, H2 and CO2. Now, the end product of second stage will act as a raw material for the third and final stage. And this is the stage where we are getting our product, that is biogas. So, we will use some of the methane producing bacteria from the table. If you remember these one, methane forming bacteria. So, if we have hydrogen or H2, as we can see that we have hydrogen as our end product here. So it will be a raw material or the raw product in the third stage. So if we want to convert hydrogen into biogas, we will use bacteria which belongs to hydrogenotrophic class, right? And second, if we have acetic acid, this acetic acid will be converted into methane and CO2. This will be function of acetoclastic microbes belonging to methane forming bacteria, right? I hope now this table makes some sense. So after this, this is, as I mentioned, this is again the brief description of the anaerobic digestion process. And uh, again, there are redundancies here because I want you to understand that the whole anaerobic digestion process is primarily divided into only three stages. One in which the complex molecule is getting converted to smaller and simpler molecules. These simpler molecules are further getting converted into acetic acid or H2. This acetic acid and H2 are then subjected to methane producing, and they produce methane and CO2, which is basically our biogas, right? Then we will now see more detailed information about each step, right? So first step was hydrolyzing and fermentation. So hydrolyzing and fermenting bacteria. The first step in the fermentation of complex substrates is the hydrolysis of polysaccharides to oligosaccharides and monosaccharides, of protein to peptides and amino acids, of triglycerides to fatty acids and glycerol, and of nucleic acid to heterocyclic nitrogen compounds, ribose, and inorganic phosphate. So, repeating myself again, hydrolysis of fermentation. These are the process in which we are converting complex molecules or complex carbohydrate, complex protein, or complex fats into their simpler form. So carbohydrate, you will convert carbohydrate into their monomer, oligomers, or saccharides. Proteins, you will convert them into either amino acids or peptides. Fat, you will convert them into either fatty acids or alcohols, depending upon the type of fat which was available in your raw material or the waste material here. Next is the relative anaerobes of genre like Streptococcus and Enterobacterium. Bacillus, Cellulomonas, Mycobacterium are some important hydrolytic bacteria. So now we have examples, right? Streptococcus, Enterococcus, they belong to the first stage here, that is hydrolysis and fermentation. Next, hydrolytic enzymes. As I mentioned, you can either use the microbe directly, or you can use the enzymes extracted from these microbes. So we know the microorganisms which are involved in hydrolytic step are Streptococcus, Enterobacterium, Bacillus, Mycobacterium, right? And some of the enzymes at this hydrolyzing step are cellulases, which will digest cellulose, hemicellulases, which will digest hemicellulose, amylases, which will digest starch, lipases, which will digest fat, and proteases, which will digest protein, right? As you can see here. So, with the help of these microbes, Streptococcus, Enterobacterium, Mycobacterium, Cellulomonas, we will be able to digest protein, fat, and carbohydrate into their constituents, that is amino acid, monosaccharides, and fatty acids, respectively. Similarly, if you want to substitute the microbes to the enzymes, then we have cellulases, hemicellulases, amylases, lipases, and proteases. So, yeah. So again, this is just for you to get a deeper understanding of what are the products of hydrolysis and fermentation. So if you're subjecting carbohydrate to hydrolysis step, either using the whole microbe, which are listed here, or subjecting these carbohydrate to enzymes, which are listed here, we will get sugars and alcohols. Next, if we are subjecting cellulose, right? So for cellulose, what we will do? Either we will subject the cellulosic raw material to cellulomonas, which will digest cellulose, or we will subject cellulose or this raw material to cellulases, which will break this cellulose. And what it will form? It will form basically glucose and cellobiose. Glucose is the monomer and cellobiose is the dimer. Proteins, it will convert it into amino acids. Peptides, again, it will convert peptides into fats, fatty acids, and glycerol. Lignin, degraded very slowly, right? So here comes the role of lignin and hemicellulose or lignocellulosic material, which we were talking about. Why it is difficult to digest lignin or lignocellulosic material? Because we don't have good microbes to digest them, right? So if we have microbes which have the machinery in them, which are able to metabolize lignin, then this is not a problem. The problem comes when we don't have the machinery or the enzymes which will digest these complex compounds. So this is the problem with lignin. Hence, if we subject it to pre-treatment, we're going to discuss it in the coming slide, that if we subject it into pre-treatment, it helps the microbes to break the compound or the complex compound more easily. So continuing with our discussion of hydrolyzing and fermenting bacteria. So obligate anaerobes include bacteroids, Bifidobacterium, Butyrolvibrio, Eubacterium, and Lactobacillus act as acidogenic bacteria, right? Many enteric coliform bacteria, classically represented by the pathogen Escherichia coli in the genus Escherichia, and pathogens in the genre Salmonella and Shigella, are also some common fermentive or acidogenic bacteria. Till now, we were talking about hydrolyzing bacteria or hydrolyzing enzymes. Now we are talking about the sub-step of the first, that is the fermentation. So for fermentation, what we can use? We can use obligate anaerobes. What are obligate anaerobes? Obligate anaerobes are the microbes which only survive in the absence of oxygen, and they do not need oxygen for their survival, right? So some of the bacteria which belong to this category of obligate anaerobes are bacteroids, Bifidobacterium, Butarovibrium, Eubacterium, Lactobacillus, right? And the classical Escherichia coli. If you know or if you do not know, it is just some fun fact to know. Escherichia coli also lives in your intestine, right? So in your intestine, there is no oxygen. It is anaerobic condition. Hence, it lives there and it helps in maintaining a good gut microbiota for you. It helps in digestion of your own food. But there are some cases in which Escherichia coli causes disease, but that strain of this microbe is not naturally present in our body. It is a strain which causes disease and it is called virulent strain, right? So not all Escherichia coli causes disease to you. Some of the Escherichia coli strain does cause severe disease to you. On the second hand, even if you have a Escherichia coli, which is a resident of your own intestine, and its number increases to the normal level, which is permissible for your gut, it causes disease, irrespective of whether it is a pathogenic or the virulent strain or not. So, just a side information. So, the products which are formed when we subject these fermentive bacteria to the raw material are listed here. So, if we subject sugar, it will convert sugar into fatty acids, carbon dioxide, and hydrogen. If we give alcohols, they will get converted into fatty acids and CO2. If we give amino acids, it will get converted into fatty acids. If we give glycerol, it will get converted into acetate of CO2. So now you can see the amount of raw material. These are simpler compounds, right? Why? Because the complex compounds, we have already subjected them to hydrolysis to convert them into simpler compounds. Now, these simpler compounds are subjected to fermentation and they are further converted into more simpler or unit or like monomeric form of the compounds, right? So now we are gradually degrading the complexity of the waste material or raw material here. Now we have these compounds as a raw material for our next stage. So what are acetogenic bacteria? As I mentioned earlier also, that acetogenic bacteria will make acetic acid, right? And some of the examples include Clostridia or Acetovibrio, right? So hydrogen producing acetogen, for example, we have propionate, we have propanol, and we have butyrate as raw material. If you subject them to acetogens, they will make acetic acid, CO2 and H2. If we give them to homoacetogens, a class of microbes, they will use CO2 and H2 and convert it into acetic acid, right? So in one, we are using simpler compounds, that is propionate, propanol, and butyrate, to get them converted into acetic acid, CO2 and H2. In the next, or homoacetogens, we are using CO2 and H2 to convert them into acetic acid and water, right? So coming to the last stage of the anaerobic digestion, that is methane production stage. So we have methanogenic bacteria. What were methanogenic bacteria? There were two methanogenic type of bacteria belonging to this category of microbes. First was osteoclast and other was hydrogeno. Oh, I'm forgetting the name. That is, yeah, hydrogenotrophics, right? So for methane producing in osteoclastic methanogen, what they will do, they will convert acetic acid into methane and CO2. For hydrogenotrophic methanogens, we will get methane by using H2 and CO2, not the acetic acid. And if you remember, acetoclastic methanogen were more than 50% efficient in converting acetic acid into biogas, while hydrogenotrophic were only 20 to 24% effective in converting hydrogen to methane, right? And it is written here also that 70% of methane is produced by this route, that is osteoclastic, by using osteoclastic methanogen, They are highly sensitive and having slowest growth rate. Next, for hydrogenotrophic methanogens, 30% of methane is produced by this route, right? And they're less sensitive and having high growth rate. So, formic acid and methanol can also be converted to methane by bacteria. These are just the raw materials which we have with us, which can be converted into methane and carbon dioxide using methane producing bacteria, methanogenic bacteria. So, this is the brief description of the methanogenic bacteria. So, methanogenic bacteria are unicellular, gram variable, strict anaerobes that do not form endospores. Their morphology, structure, and biochemical makeup are quite diverse. More than 10 different genres have been described. The methanogens have been divided into three groups based on the fingerprinting of their 16S ribosomal RNA, or in short called rRNA, and the substrates used for growth and methanogenesis, right? So, what kind of bacteria are methanogens? They are unicellular, that is, they are single cell. Gram variable. Gram variable means that we have a staining technique called gram staining. It divides microbes into two categories, that is, gram positive bacteria and gram negative bacteria. The bacteria which are able to retain the color of the original strain or the gram stain are called gram positive, and the bacteria which lose the color of their original stain are called gram negative. But this classification is not useful for methanogenic bacteria. Why? Because in this group of bacteria, that is methane producing bacteria, they have both gram positive as well as gram negative bacteria, right? Hence, they are called gram variable. Next are strict anaerobes or obligate anaerobes, as we have discussed earlier, that they will not survive in the oxygen rich condition. They need the absence of oxygen for their survival and for their optimal growth. And they do not form endospores. Endospores are basically their dehydrated form of the bacteria, which helps the bacteria in surviving some harsh conditions, right? So what are methanogens? They are the bacteria which are of unicellular type. They are gram variable and they are obligate anaerobes, right? Their morphology, structure, and biochemical makeup are quite diverse. More than 10 different genres have been described, meaning within the methanogenic bacteria, there are 10 different subgenres of this bacteria. And how we are deciding whether this belongs to whether a microbe belongs to methanogen or methanogenic bacteria, we use the fingerprint of 16S rRNA. We're not going to discuss too much about this, but just know this, that we all have RNA and DNA as our genetic material, and throughout the life forms from prokaryote to eukaryote, 16S RNA is a type of RNA which is found in the ribosome. This type of RNA is highly conserved. So, if an organism belongs to one particular class of classification, more or less they have same or similar 16S rRNA signature. Hence, we can classify microbes based on their 16S rRNA fingerprint, right? So, apart from the 10 different genres, here we are discussing some three groups which are in general present in methanogenic bacteria. Group 1 has Methanobacterium and Methanobreviibacter type of microbes. Group 2 has Methanococcus, and Group 3 has several genera including Methanomicrobium, Methanogenum, Methanospirillum, and Methanosarcina. You do not need to remember the exact and remember all of the microbes. You just need to know that, for example, in group 1, Methanobacterium type of microbes are found. into coccus or methanococcus type of microbes are found, while group 3, they have diverse a number of or diversification in the methanogenic bacteria. Now, the fermentation process, right? So fermentation is a very elaborative process. So the pathways for fermentation, as you know, that in fermentation we were using what? We were using the products of hydrolysis to convert the simpler constituents into their more simpler forms, right? The pathway which does this, or the reaction or the mechanism for the fermentation, was given by three scientists, on which the pathway is named, that is EMP pathway, the Embeden, Meinhof, and Parnas, right? So these three were responsible for the production of pyruvate. They defined the fermentation process, and the EMP pathway, excuse me, is responsible for the production of pyruvate from sugar as an intermediate product, which is further converted into acetate, fatty acids, carbon dioxide, and oxygen, right? Sorry, and hydrogen. So at low partial pressure of hydrogen, acetate is favored. At higher partial pressure, propionate, butyrate, ethanol, and lactate are favored, generally in that order. So what we mean by this? See, there is a pathway, right? So in this pathway, we have pyruvate from sugar. I don't know whether most of you are aware of glucose or glycolysis, which is basically the first metabolic pathway used by your body to convert glucose, the food which you are having, into pyruvate, right? Pyruvate further has multiple roles. During the aerobic cycle, it gets converted into acetyl coenzyme A, and other went into Krebs cycle and all that stuff. It will give you energy at the end of the day. Hence, from these pathway, your energy is coming. If we eat food, we feel energetic. So this energy is coming from converting glucose to pyruvate and further pyruvate into ATP and energy via Krebs cycle during aerobic, or the during when oxygen is available. But when oxygen is not available, hence we are talking about the fermentation process, right? And we are talking about the process which are taking place inside the microbes. So the pyruvate, instead of going into Krebs cycle, which is a part of aerobic pathway, it has multiple routes to get converted based upon the partial pressure of hydrogen in the system. This is one type of switch. So if we have low partial pressure of hydrogen, the pyruvate will predominantly get converted into acetate. And this acetate, in our later stages, we will make biogas, right? At higher partial pressure, this pyruvate will get converted into other type of products, such as propionate, butyrate, ethanol, and lactate. As we have seen in the previous slide, the propionate, butyrate, they also can be converted into methane, right? If we use the right acetogenic bacteria. So this propionate, from where it is coming from? It is coming from the system when the partial pressure of the hydrogen was less, and the pyruvate was getting converted into propionate instead of acetate, right? I hope now you are able to connect the dots. So if we have propionate butyrate, it is not that we are not able to produce any biogas because we don't have acetate. From acetate, we know a direct path that we can convert it into methane. For propionate and butyrate also, we can convert these into biogas or methane at the end of the day. So there is also a special mode of cleavage of intermediate pyruvic acid to formic acid by enteric bacteria that is not found in other bacterial fermentations, right? You do not need to worry about formic acid, but you should know that during fermentation, the products of hydrolysis are getting converted into simpler compounds, and the formation of these simpler compounds depends upon the partial pressure of hydrogen. If the pressure is low, acetate will be favored. If the pressure is high, propionate, butyrate, and other small weak acids will be favored. right? So when we were talking about lignin and we said that they are very hard to digest by the microbes because we don't have microbes, right? So we need to help these microbes to digest better. How we can help them? We can help them by providing them some simpler units or some simpler form of lignocellulose. So how can we give simpler form of lignocellulose? By subjecting it into pre-treatment. That pre-treatment has a different category. For example, we can give physical treatment or pre-treatment, we can give chemical and physico-chemical pre-treatment, and we can give biological pre-treatment. In the physical pre-treatment, the process which we are subjecting our raw material, which is high in lignocellulose or lignin constituent, we either subject it to milling, that is, we are making it into smaller sized particles of the raw material or waste material. We are subjecting the raw material or the waste material to irradiation and other methods such as hydrothermal or pyrolysis, right? And what we can expect if we are giving such treatments to or such physical pre-treatment to our waste material. First, it will increase the accessible surface area, hence it will be easier for the microbe to work on the compound. It will decrease the cellulose because it will break down the cellulose. Next, it will decrease the degree of polymerization. That is, again, it will help in getting the microbe the simpler form of lignin or lignocellulosic compounds. Some notable remarks that most of the methods are highly energy demanding. Most of them cannot remove lignin and preferably not used for industrial applications. They are more for studying purposes and lab scale studies. For chemical and physico-chemical pretreatment, we can use explosion of CO2 and SO2. We can use alkali. We can use acids. We can use, again, gas and some oxidizing agents, right? So what they will do for the lignin material, they will increase the accessible surface area. They will partially or nearly complete delignify the material. They decrease the cellulose. They decrease the degree of polymerization and also partial or complete hydrolysis of hemicellulose. Hence, the material which we will subjecting our raw material now after pre-treatment to the microbe will be low in cellulose and will be the simpler form of hemicellulose, right? This was the whole sole purpose of subjecting our raw material to hydrolysis to convert complex into simpler forms. Hence, for lignocellulosic material, providing pre-treatment, it will give us the similar results. Next, for the biological pre-treatment, we can use some fungi and actinomycetes. These are basically fungi only, belongs to fungi. So if we use certain fungi, they help in delignifying the material. They reduce the degree of polymerization and partial hydrolysis of hemicellulose, right? So again, these are reducing the complexity of the lignin, and then we can subject this for the anaerobic digestion processes, right? So remarks for biological pre-treatments are low energy requirement, obviously because we are using microbes. Chemicals are not required. Mild environmental conditions, we do not need to provide some high acidic or high temperature conditions. Very low temperature treatment rate. Did not consider for commercial applications. Obviously, again, these are all for study purposes and lab scale studies only. With this brief introduction of anaerobic digestion, now I would like to discuss the previous year questions with you guys, and we will see how to solve them, and they will help you in solving your current assignment. So as you know, I will give you each for numericals 5 minutes, and for easier question ranging from 1 minute to 2 minutes. So I will need some active participation from you guys. So here comes our first question. So in anaerobic digestion method, normally the dash is a rate limiting step if cellulose content is higher in feedstock, and dash may be rate limiting step. Though we have not discussed it, but I hope from a brief discussion you can even guess if you do not remember the exact mechanism. So is it first the hydrolysis, methanogenesis, second methanogenesis, acidogenesis, third acidogenesis, hydrolysis, or methanogenesis and hydrolysis? I will give you guys 2 minutes for this and just give some answer in the chat box only. I hope you guys were able to understand the question and able to answer. So if your answer was methanogenesis and hydrolysis, then your answer is correct. We will see that during this discussion of methane production kinetics, as it was having multiple steps, which we have even discussed today, there are some of the steps which are rate limiting depending upon the raw material which we have. So in general, methanogenesis or the last step of the biogas production is the rate limiting step. But if the cellulose content is higher in the feedstock, hydrolysis may be the rate limiting step. Why? Because now we have discussed that if the material is high in lignin or lignocellulosic material, we need to pre-treat it and then subject it to the hydrolysis and fermentation. So in normal conditions, the raw material which we are subjecting to the hydrolysis and fermentative step is not that complex in comparison to cellulose or lignocellulosic constituent. Hence, if the material is rich in cellulose or lignin or lignocellulosic material, the hydrolysis step will be the rate limiting step, right? Now for second question, I will give you each 1 minute for each question, and then we will discuss the answer. So the second question says, the type of losses in microbial fuel cell caused by uneven depletion of reagents in electrolyte is called as. If you remember the lecture, microbial fuel cells are the cells which we are using nowadays, and it is quite a new field of research. So there were certain losses associated with this process. So here we are asking about a particular loss which is associated with the depletion of or uneven depletion of reagents in the electrolyte. Is it ohmic loss, activation loss, bacterial metabolic loss, or concentration loss? Even if you do not remember the exact name of the loss, you can guess from the question itself that what we are talking about. Your 1 minute start now. So, if you answered concentration loss, then you are correct. So, obviously, even if you do not remember it, ohmic loss will be due to the loss in the current, activation loss will be due to the loss in the uh, uh, initiation of the process, and bacterial metabolic loss will be due to the losses which are incurred during the metabolic process, and concentration loss are the one which are due to the uneven depletion of the reagents. If you do not remember even, then you can go back to the lectures which Professor Mandal has discussed, and you will get the exact thing there also. So they were ohmic losses. Activation losses are because of accumulation of gases or other non-reagent products, right, at the interface between electrode and electrolyte. Bacterial metabolic losses, as I mentioned, it is due to the metabolism of the bacteria itself. Then concentration loss. This is because of the uneven depletion of reagents in the electrolyte, which causes concentration gradient in boundary layer, right? And next one is the mass transfer to the membrane. And last is the voltage loss due to pH difference between cathodic and anodic chamber. I will highly recommend you to go back to the lectures if you're not able to answer some direct questions, and it will help you in remembering all the things which were discussed during those lectures. So our next question is uh, theoretical substrate conversion rate per unit reactor volume can be estimated by. So even this is a direct question. I will give you guys 1 minute. If you do not remember, just try to derive the formula by knowing what each variable here means, and you will be able to get the answer. I'll give you 1 minute now. So if you answered option number second, then you are correct. If you do not remember from this, I have given the exact slide where it was discussed. So the theoretical substrate conversion rate per unit reactor volume can be estimated as the following. That's R is equals to S naught mu max theta minus 1 minus Kx upon theta mu max theta minus 1, where R is the substrate converted per liquid volume at hydraulic retention time theta. S naught is the substrate concentration in the feed, that is the initial concentration. Mu max is the maximum specific growth rate of the microbe. And Ks is the saturation constant or substrate concentration at which the specific growth rate is half its maximum specific growth rate, right? So, in general, as I mentioned, that microbes require particular conditions for their growth. So, this particular formula at 30 to 37 degrees Celsius, we have observed that with the help of this operation, there is optimum conversion of glucose, and theta can be achieved for 4 hours and 4 days in the acid and methane phase reactors respectively, right? So everything or the every yield of a microbial route or the biological route will depend upon the conditions in which the operation is being conducted. This was not the case with the thermochemical processes, right? So we need to be extra careful about the conditions in which we are using our anaerobic digester or or any biological route process. Coming to our next question, even this is a simple question. So, which of the following has highest C to N ratio? Is it dry rice straw, dry wheat straw, corn stalks, or fallen leaves? If you do not remember the exact number, I think you can make a pretty good guess depending upon the raw materials which are given here. So I will give you guys 1 minute and then we'll discuss this. So if your answer was dry wheat straw, then you are correct. So if you not remember the exact ratios, I will recommend you to go back to this slide and see. Do not remember every single detail in this slide because obviously it is not practically possible for you guys to know everything at top of your head. But just remember or try to keep in mind the highest and the lowest CN ratio materials, right? So here we talked about the highest CN ratio. We can see that the dry wheat straw has 87 is to 1 ratio of carbon to nitrogen, right? And the lowest can be fresh pig manure. which is literally 13 is to 1, right? So just remember the highest and the lowest, and you do not need to worry about the all the numbers on the slide, right? We'll go to our next question. So our next question is, it's a picture question. So in this, you need to identify the different zones in this anaerobic digester. So there are three zones given here, A, B, and C. You need to tell me the zones which are here. Is it first, A is sludge zone, B is mixing zone, or C is fluid zone? Or A is fluid zone, B is mixing zone, and C is sludge zone. C is A is fluid zone, B is sludge zone, or C is mixing zone. Last is A is mixing zone, B is fluid zone, and C is sludge zone. Again, even if you do not remember the exact diagram of the anaerobic digester, with some pre-understanding of how the process was taking place, you can easily get the answer. Though remembering this or knowing the components of a digester will help you in understanding the process at the end, right? So I will give you 1 minute, and then we'll discuss the answer for this question. So if you have answered option number C, that is A is fluid zone, B is sludge zone, and C is mixing zone, then you are correct. And if you are not able to remember, you can go back to the lectures of Professor Mondal, and he has given a very nice explanation about all the areas or all the regions present in the anaerobic digester and what are their functions and roles. So if you understand their function and role, you will be able to understand the zones itself, and then you do not need to memorize the zones which are present here, right? So I will recommend you, if you are not able to answer this, I will recommend you to go back to the lectures once and then again go back to such questions and try to understand and get the answer directly. Our next question is also a picture question. So here also you need to identify the missing components. Identify A, B, and C in the fuel cell, right? So it's a diagram of a fuel cell. So is A is anode, B cathode, and C is proton exchange membrane, or A is cathode, B is anode, and C is proton exchange membrane, or A is anode, B is cathode, and C is rigid wall, or I apologize for the connection problem. We will get back to our discussion in a while. I'll just share my screen with you. In a minute. Yeah, I hope you guys can see my screen, right? Yeah, so we were discussing about this. I think there is some technical issue from my side. I apologize for this. We will try to wrap this up soon so that uh, we do not have any such inconvenience. Yeah. So the first is A is anode, B is cathode, and C is proton exchange. Even if you do not remember that what was anode and what was cathode, you can remember that from just go back to the slides and it will help you out. Even if you are not able to understand it, the basic concept is that on anode, uh, there is a particular reaction which occurs, right? In this, we use the hydrogen, it is getting excuse me. Yeah, so go back to these lectures, and if you are not able to understand this, we can discuss this in the coming Saturday class. It will be on regular day and on regular time, right? Yeah. Then our next question is the relation between total digester volume Vt and the total sludge volume Vs is given by. It was a very direct relation, that is the total volume is equal to twice the sludge volume. If you do not remember it, this was the part of the slides which was discussed in the lectures. And here you can see that the end relation between the sludge volume and the total volume is twice. That is, the total volume is twice the sludge volume. Now, this is the numerical type of question. So I will give you 5 minutes for these questions. And this type of question was discussed in the lecture. So I hope you guys are able to attempt it. And even if the answer is wrong, just try to attempt it and try to understand what the question is asking and how to solve it. And obviously, we will discuss this. We will discuss the solution here also. So the question states that the volume of the digester chamber required for the biogas production from the cow dung of 12 cows having body weight of 180 kg each, right? Assume the temperature is 30 degrees Celsius. The solid content of the cow dung is 15% and hydraulic retention time is 45 days. One cow produces 10 kg cow dung daily, right? So what we need to know? We need to know the volume of the digester. Try to attempt it and we will discuss the solution. I will give your 5 minutes. I will give you 5 minutes and your 5 minutes start now. I hope that you were able to attempt it. And even if not, no worries, we'll discuss it here. So the answer is 12.65 meter cube. So to those who have got the right answer, congrats to you. And to those who are not able to get the answer, don't worry, we will discuss how to proceed with this. And I will recommend you to again go back to this particular lecture because there it was very nicely explained how to approach such problem and what are the steps required for solving such questions, right? So the solution is pretty easy. So the total discharge will be 10 into 12 because we have 12 cows, right? And each cow produce 10 kg of cow dung easily. So the total discharge will be 120 kg. Now the total solid of the fresh discharge will be, as it is mentioned, that 15% of the cow dung uh, is used, right? Or is used for the slurry and the anaerobic digestion process. So it will be, so the 15% of 120 will be 18 kgs, right? Now we know that for favorable conditions in the digestion process, we use 8% concentration. So 8% means 8 kg of solid and 100 kg of influent. But we have 18 kg of solid, one such solid, right? We have 120 kg of one such solid, which has 18 kg of total solid for of fresh discharge. So how we can do this? We can basically take the 8% concentration for the 18 kg solid, which will come around 225 kg of influent. Correct? Now, this 225 kg of influent can be subjected to the digester, and the volume or the working volume of the digester is basically 225, that is the influent concentration or the influent quantity into the retention time, that for how much time we are subjecting this solid, right? It is 45 days given. So we have 101.25 kg. Right? Now you have to convert this kg into meter cube. So you will get 10.125. So this is basically the working volume of the digester. Correct? So this is the working volume. But for geometrical assumptions, I will show you the slide in which these assumptions were made. And you need to know this for solving such questions. There is no shortcut for these type of questions. So the working volume is basically the 0.8 of total volume, right? Or the digester volume. So from this, we can easily get the total volume or the volume of the digester chamber by using the relation 0.8V, and hence we will get 12.65. Don't worry if you do not remember this. This is the slide where every relation was given. So we have used this, the total volume or the VGST or VD here. These were discussed in the prior slide to this particular slide. You can get to them. So this volume is basically 0.8 times of the digester volume, right? So for such type of questions, there were two assumptions. First, which you need to know, is that 8% is the operational condition for these type of digestion or anaerobic digestion processes. And second, these relations, right? That the volume, how the volume is related to the total volume, how the diameter is related to the total volume. All these relations you need to know for solving such questions. There is no shortcut, I'm sorry to say. Yeah. So we'll move to our next question. Again, this is a numerical type of question. In this problem, that is problem number 8, the diameter of digester is, right? So even in this question, you need to know a relation of volume and a digester. So you know the volume, and if you do not remember, the volume was 12.65 meter cube. This is the volume. So from the given volume, you need to calculate the digester diameter. So there is no direct formula, but there is a direct relation which you need to remember. So I'll give you 5 minutes for solving this question. It is a small question. If you know the relation, then you can easily solve it. But if you do not know the relation, you need to put some mind in it for solving this question. So though it's a short question, so I will give you only 3 minutes for this, and then we'll discuss the solution, right? For your purpose, I'll will show you the relations here. So try to use these relations, right, in getting the diameter, right, from the volume. And the volume, again, I'm repeating, the volume is 12.65 meter cube, and you need the diameter in meter itself. And here are the relations. So I'll give you 3 minutes, and your 3 minutes start now. I hope that you guys were able to attempt it. It's a pretty straightforward question. So if your answer was 3.047 meter, it is the right answer. And the relation which we have used here is D is equal to 1.3078 volume to the cube root. So it is basically this relation between the diameter and the volume, right? So if you put the values in this, you will get the answer directly. Coming to our last question, again, it's a straightforward question, and even if you do not remember the exact formula, so in this case, it will affect you because you need to know the exact constant which are ahead of the variable terms, right? So you need to remember this formula. So what it is asking, it is saying the major proportion of cellulose and hemicellulose are glucan and xylem respectively. Digestibility of both glucan and xylem can be determined from the yield of their respective monomer and respective monomers and disaccharides. The digestibility of xylem can be given as. So you need to know the direct formula. I hope that you are able to remember it, but even if you are not, so no worries about it. You can always go back to the lectures and uh, remind yourself what was the formula and the story behind getting these formulas. So I'll give you each 1 minute as it is a theoretical question, not a numerical question, and then we will discuss the right answer. Yeah, the time starts now. Yeah, so without further ado, we can look at the answer, and the answer is amount of xylose in grams plus 1.064 times amount of xylobiose in grams divided by 1.136 times the amount of xylene in grams into 100 for the percentage, right? Even if you do not remember it, this was the slide in which it was discussed, right? You can directly see here that the digestibility of glucan and xylene are given here. So in this, we asked only for the xylene percentage digestibility. We have glucan percentage digestibility also. So for xylene, the products are xylose and xylobiose and xylene, while for the glucan, the variables will be the glucose, cellobiose, and the glucan itself, right? So don't worry if you are not getting these questions, and if you are getting it, congratulations, you are having a good preparation for your coming exam. And to those who are not able to get this, don't worry about it. The lectures are there with you. You can anytime you can go back to those lectures. If you have any doubts, we can discuss in the next class. And also, you know that we have a Sunday class also with for the same topic, right? So due to some technical difficulties, I was not able to take the class this time. But we have our regular class next Saturday from 3 to 5 p.m. There is no change in that schedule. And I will recommend you to go to the lectures once after discussing in the live session. It will help you in remembering all the old things, and you do not need to then sit separately for preparing specifically for the exam of this subject. You can easily pass through the exam if you are attending these live sessions, right? So I would like to thank you all for your patience listening. And if you have any queries, I have my website link in the first page of the slide. You can always send me a mail, or you can put questions in the discussion box of the course also. So thank you, and we will see each other uh, in the coming Saturday. Thank you.